Welcome back to SBT 408 Toxicology. We are continuing today with toxicokinetics, one of the major processes that is talked about in the field of toxicology. We did speak about it briefly during our introduction to the course on the first day. We did talk about toxicokinetics and toxicodynamics. Today we're going much into toxicokinetics, which you would come across even not only in toxicology but also in pharmacology, and you will usually hear this process being referred to with an acronym ADME, where A means absorption, D means distribution, M means metabolism, and E means excretion. And it's a major thing you come across all the time because this speaks about what the body does to any xenobiotic that enters it. These four major processes do occur. So what you see on your screen now is a depiction of the stages that a xenobiotic undergoes in terms of exposure, first of all, the organism. So if you take, say, this rectangle as an organism and the external part of the rectangle as its environment, a xenobiotic is coming from the environment, from the external part of the organism into it. And so you have an exposure of the organism to that substance, first of all. And once the substance gets into the organism, then the processes of absorption, first of all, occurs, which is, absorption into, say, the bloodstream. And then you have, of course, that is when you have the substance getting into the organism, mainly by oral ingestion, or even maybe through skin, derma, because in some cases, some of these substances deliberately can be injected, say, intravenously. That would be direct, which means that immediately there will not be that process of absorption, but it will quickly go into distribution. Now, in distribution, you can have the substance in the bloodstream binding to some blood proteins, some plasma proteins, and in so doing, in binding can, have some effect. Or the substance, even in the bloodstream, goes to bind to some specific receptors in some tissues, some tissue cells, and then will have its toxic effect. Or you can have the substance, first of all, getting distributed to, various tissues, various organs, and then that substance undergoes a process of metabolism. What is being referred to as biotransformation is the same as metabolism of the substance. And then you have the metabolites of the substance, then having the toxic effect. Here again, you can have, say, straight from distribution, the substance directly will have an effect. Not the metabolite, but the substance directly is having an effect after getting distributed in the various organs and tissues. So what you notice here is that it is either the substance is having a direct effect on a target cell, on a target tissue, or if you take the horizontal pathway, the substance binds to some proteins, which means to form some complex with some proteins, and it is that complex which will then be having the toxic effect within the organism. And then in the last hand, after all the effect, of course, when all that is happening, the substance is the organism is also having this process of metabolism and trying to remove the substance from the body through, say, mostly the process of biotransformation, because some of the substance is going to be excreted unmetabolized. So substances, xenobiotics that enter into the body can either get excreted as metabolites, or they get excreted as unmetabolized substances. So that is going to be the very last process, excretion or elimination of the substance out of the body. So what we'll do is we'll take a look at each of these stages: absorption, distribution, metabolism, and excretion. We take each of them and go through the process, looking at different mechanisms, different processes, and even take a look at some specific examples. A lot of times these processes have been studied with medicinal substances, primarily because for medicinal substances, because these are developed drugs for some therapeutic purposes, and they have to be studied for their toxicological effects and all of that, clinical trials and all of those things. So those medicinal substances have much more information as far as the toxicokinetics is concerned, rather than the what we refer to as, say, toxicants. So a lot of times when we talk about toxicokinetics, you realize that the conversation shifts a little bit into pharmacokinetics, except that the focus is on toxic effects of the substances rather than the pharmacological or therapeutic effects of these substances. You remember from the beginning I mentioned that the field of toxicology actually sort of emanated from pharmacology, and because of the historical background, we did see some of these histories of how some of the medicines caused a lot of problems, especially in the 50s and 60s, which resulted into the development of the field of toxicology. And so you see that in the examples that we'll be using, some specific examples, it will be very much related to medicinal substances to understand some of these processes. Now, to take a general overview of the pathways of xenobiotics in the body. First of all, you have any xenobiotic will get into biological system through a route of entry. So you have a major route of entry of xenobiotics is by oral entry. So you have substances that get into the body through the mouth, the buccal cavity, and through from the buccal cavity, the mouth gets into the digestive system, that is the gastrointestinal tract. Now, you know what happens in a gastrointestinal tract. You have a metabolism of food. But these are the xenobiotics we talk about, of course, do not go through that metabolism as food undergoes in the gastrointestinal tract, but they get absorbed together with the nutrients from the food into the bloodstream. So, in the digestive system, you have absorption of the nutrients in the small intestines into the bloodstream. And when that absorption is ongoing, you have the xenobiotics also getting absorbed into the bloodstream. Now, once the xenobiotic is in the bloodstream, it flows to organs, And particularly, we have metabolism of xenobiotics occurring in the liver. And so the substance gets to the liver, and the substance then undergoes metabolism, then its metabolites, and some of the substance that don't get metabolized also, gets into bile. So these are like waste products, that become stored in the gallbladder. Now, from the gallbladder, they get secreted back into the gastrointestinal tract in the duodenum, because bile is secreted into the duodenum to emulsify lipids. And so in that bile, you have, these xenobiotic metabolites as well. And so in the end, these metabolites and some unmetabolized part of the xenobiotic gets excreted through stool, through fecal matter. You have these substances getting excreted. So that's one pathway that a xenobiotic can undergo. You can have xenobiotic also undergoing, going through another pathway, which is through inhalation. So through the nasal cavity, a substance, so for gaseous substances, gaseous xenobiotics, they are inhaled through the nasal cavity. And once they are inhaled, they get into the lungs. So that's going to be, first organ there that is going to be having that process of absorption there in the lungs. Because if you take a look at the structure of the lungs, that's an organ with lots of blood capillaries, small pockets, what we refer to as alveoli. And so the substance gets absorbed into the alveoli. And of course, also, the process of inhalation of gases, say oxygen, and oxygen goes to the tissues and carries out, is involved in the metabolic processes, and you have the byproduct, carbon dioxide, coming back to the nasal cavity, back to the lungs, and then being exhaled out of the body again through the nasal cavity. So in that exhaled air, which is coming out, you do have the metabolites of xenobiotics also being exhaled. You have exhalation of some aldehydes. So for instance, alcohol consumption, which the metabolism of alcohol results into some byproducts like acetaldehyde, for instance, which is exhaled, and in persons who are heavy drinkers, for instance, if you go close to them, their breath smells like acetone because of lots of this acetaldehyde that is getting exhaled, together with the exhaled air. So that's another pathway through inhalation, and then the byproduct, or that last process of excretion is through exhaled air. Now, if it is by intravenous entry, so that is mostly going to be say the use of medicinal substances. Of course, there are some illicit drugs that some persons also get into their systems by intravenous entry. Those who are those, some people, drug addicts, making use of some narcotic drugs make use of this route of entry as well. That's some form of intoxication, and such route of entry results into the substance getting into, say, the blood lymph, and you have some extracellular fluid so because the blood lymph is extracellular, so you have the extracellular fluid also having that substance getting into it. But from the blood lymph, you have lymph going into the kidneys, and in the kidneys, you do have some process of filtration, and then you have storage of urine together with the metabolites, and the unmetabolized part of the drug getting stored in the bladder, the urinary bladder, and later you have the xenobiotic getting excreted with urine. Or from the extracellular fluid, you can have some exocrine glands, say of the skin, like sweat glands, that secrete metabolites or some of this drug through sweat. So some part of xenobiotics also get excreted through sweat. You have in the extracellular fluid, some cells also get absorb these substances. I mean, particularly in what we refer to as soft tissues. Those are like the muscle tissues, and some of it gets into the bones. So you see, it's like this is like some form of accumulation, storage of substances in the bone. That I think I did mention that in some cases, you have substances that get stored, like, say, maybe in the adipose tissue, the very last one there. And some of it also gets stored in bones. And as if the substance is not there, and later in old age, when people begin to lose weight, these substances get released back into the bloodstream and begin to cause cumulative effects, serious effects for such persons in old age. When they begin to lose bone mass, and they begin also to lose weight in general, and the adipose tissue under the skin begins to break down, you have these substances getting released back into the bloodstream, which causes some serious health problems for persons in their old age, when actually the exposure to the substances were when they were younger. But in all of this, the major pathway of the excretion of most xenobiotics is through urine. That is why you see a lot of chemical analytical methods that test for substances in biological samples. The samples that are used for testing of these things is urine. That is if you want to make use of a non-invasive method in testing for these things, then urine is the most sample which is used. Of course, all of these samples, I mean, below here, like stool, in some cases. So, for instance, exhaled air. You know that when drivers are being tested for alcohol consumption, of course, it is their exhaled air that is tested. So that's a sample exhaled air which is tested. So I did mention that, of course, for persons that do heavy drinking, there will be some accumulation of acetaldehyde. So you see, that sensor that is sensing acetaldehyde in exhaled air to confirm that a person is drink driving. So that is a practical example for you. So apart from, say, urine that I mentioned as the major sample, which is used for this chemical analysis. Yes, stool is used, exhaled air is used in some cases. Yes, sweat. Of course, soft tissues like so this will be more in medical settings where some tissues, especially maybe in autopsy samples, for instance, the use is made of soft tissues coming from organs. In some cases, some bones are also used, but those are very medical serious cases which they have to go deep down making use of these bones to test for maybe possible intoxication. If, for instance, maybe the death of somebody, there is a suspicion that there was some poisoning, and various parts of the various samples, various parts of the body have to be tested to confirm. So, for instance, the particular substance is not found in blood, it's not found in urine, it's not found in the soft tissues, but perhaps it's gone into storage in the bones, things like that. So those things are being used sometimes to test for possible intoxication of such persons. All right. Now, if you think of the body surfaces that have direct exposure to toxicants, you want to think of the surface area of the body part which is having contact, having such an exposure to the substance, because the surface area of the particular part of the body that is having exposure to the toxicants plays a major role on how to the extent of intoxication, because of course logically the bigger the surface area that is having contact, the more absorption the substance is going to be, which means the more toxic effects is going to be observed. And in looking at these things, you realize that the small intestine is a folded region. You remember the small intestine from your secondary school biology. The small intestine is folded into some small projections called villi, which are responsible for absorption of nutrients. So the same way, if you're having the small intestine, that is having such high surface area, 100 meters squared, that's a very large surface area to absorb a lot of substance at once into the bloodstream. And so that's where a lot of the absorption occurs. In the lungs, which follows these small intestines, you have a surface area of about 70 meters squared. And there too is because you have these small pockets, what we refer to as the alveoli. There too you have large surface area there to absorb substances. And then the skin, a major part of the body that is having exposure to the external environment is the skin. But I see if you compare the skin to, say, the small intestine or the lungs, you see the skin is just flat. It's flat. It's not folded, so that you don't have the surface area increased by the kind of folding which is in the small intestine or in the lungs. So you can notice how very, very large surface area it is that a small intestine has because of the various foldings into this villi that it has. I mean, that is an organ which is very much adapted to absorption of substances. So you have the skin having just between 1.2 to 2 meters squared of surface area to have exposure to toxic substances. The large intestine is even having lesser amount because there a lot of absorption doesn't take place. The only absorption that you talk about mostly there is reabsorption of water in the process of digestion, and then you have stool just getting excreted out from there. The stomach is also not a place for absorption. It's mostly for just contraction, relaxation of the muscles of the stomach and the food, mixing, breaking it down into smaller pieces. That's all what happens there. And so you have it also having a smaller surface area. The rectum, already getting much closer to excretion by fecal matter, is having much less surface area. The oral cavity, the mouth, is also having a small surface area, not that big. And the nasal cavity here is the least among all the rest. That is a surface area for absorption, or say getting exposure to xenobiotics. Right. So of course, in all of these, the main routes of xenobiotic entry into the body is by inhalation. That is by through the nasal cavity, and that's going to be for substances that are in the gaseous state. Ingestion, of course, ingestion should be the first because many substances get into the body through the mouth. It can be waterborne, it can be foodborne, even lots of medications are taken by ingestion than any other route of entry into the body. And then dermal, I mean substances that get absorbed through the skin. All right, so Akbelu Christopher says, what about the hair? Okay, so what you notice is that a lot of times when dermal is mentioned, it is actually mentioned with a mind that the hair is part of the skin system. The skin is also looked at as a system. It's referred to as a integumentary system, which is, so a lot of times the skin is talked about as the skin and its appendages, the skin and its appendages. And when it's said like that, it means not just the skin, but the hair is part of it, and also the nails. So the nails, the skin, the hair is taken into consideration when dermal is talked about. When you look at the skin as a system, the integumentary system. So Christopher, I hope that is okay. All right. Now, and again, not only absorption, like if you like the hair that you talk about, not only in terms of absorption of the substance, but even in terms of excretion. So some substances also get stored in the hair, for instance. Just like you have substances getting stored in the adipose tissue. That's why sometimes some toxicological analysis is also done on the hair So, Christopher, I mean, it goes both ways, not only for absorption, but also even for excretion, which will be like storage of, some toxic substance in the hair as well. Right. Now we go on to look at some factors that influence xenobiotic absorption. And what we have here is a cell. I mean, the very first part there, we have a cell that, the cell membrane is made up of a lipid bilayer. And so inside of the cell, you're having some xenobiotics that are able to, penetrate through the cell membrane into the cytoplasm. That's why you're saying there that intracellular environment, and you have these xenobiotics being shown over here. Now, a major factor that allows for xenobiotics to be able to penetrate through the lipid bilayer is number one, the size of the substance. So the molecular size of the substance should be such that it will be able to pass through those, some pores, or through directly through the lipid bilayer into the cell, by processes of, say, diffusion, for instance, like, say, maybe passive diffusion like that. It can easily pass through the lipid bilayer into it. Another is what is being referred to as lipophilicity. That is to say that the substance dissolves in lipids. In other words, that substance is hydrophobic, because the lipid bilayer is consists of what? Phospholipids. And so if the substance is hydrophobic, then it can easily pass through the lipid bilayer into the cell. Then also process of say ionization substances. When we come to the next one, we will see that substances that are having some strong charges on them, have difficulty passing through. So we'll leave that ionization and go to the next, which is the fact that, yes, molecules can also penetrate through, say, maybe intercellular spaces, and can easily get into, the cell through there. These are through some water-filled channels. Those intercellular spaces can be some water-filled channels that smaller molecules, again, but this also depends on the size. So again, that is size being talked about. So size, lipophilicity, and then charges, that is talking about ionization. So because the lipid bilayer is having a hydrophilic head, which is consisting of phosphates. Now that phosphates also gets ionized, so that if you are having a substance, a xenobiotic, that is also getting, that is also having those, strong charges, is strongly charged, then of course it's not able to pass through the lipid bilayer into the cell. Again, if the substance is hydrophilic, if the substance is too hydrophilic, it will not be able to pass through a lipid bilayer, because a lipid bilayer will rather, solubilize lipophilic or hydrophobic substances. From the very first property that we talked about, lipophilicity, that is related to the fact that if a substance is too hydrophilic, it cannot pass through the cell membrane into the cell. Or again, size. So if the substance is too large with molecular size, it cannot pass through. the membrane into the cell. You have also some membrane proteins, which are purposely for transport of substances. So you have molecules that serve as substrate to these membrane proteins, so that these transmembrane proteins they allow for some substances to pass through them and some substances not to pass through them. And it's actually in both ways. You have membrane proteins which are for transport of substances from the extracellular space into the intracellular space, and vice versa. You have some of them transporting in the opposite direction. And whilst in the cell, then you have the substance having its biological, toxic effects, biological or toxic effects in the cell. But of course, it's not all substances that have to get into the cell before they have their biological or toxic effects. You do have substances. If you remember your biochemistry. I think one of the biochemistries you did, you would have talked. I don't know if we did look at it in general biochemistry, where we talk about receptors, and you have some receptors that are on the surface of the cell, so that xenobiotics can just bind on those receptors on the cell and have their effect. And you have some receptors that are inside of the cell, and you have substances that will have to pass through the membrane into the cell to bind to those receptors before you have the effect coming out of them. All right, so Alexander says, what is the difference between ingestion in terms of toxicology? Okay, the two things, injection and ingestion. All right, so injection means you are injecting into the bloodstream, like you take injection, say intravenous injection. And ingestion means you are taking the substance through the mouth. The second word, ing, is taking the substance through the mouth. That is ingestion. And injection is when you use a needle and syringe to inject the substance. All right. Now, say we did talk about this lipophilicity, hydrophilicity. A lot of times in pharmacology, to some extent, of course, also in toxicology, to predict the permeability of xenobiotics of a membrane. That is to say, if you want to see if to what degree a substance will be able to penetrate through cells, knowing that the cell is having a lipid bilayer. You have a biphasic system which is used experimentally to test for this thing. And that biphasic system is just a mixture of water and octanol. So this the alcohol octanol is hydrophobic. It's just like an oil. So this biphasic system, the octanol water system, is a standard system which is used to test for the lipophilicity or hydrophilicity of substances. So that if you have this a mixture of water and octanol, of course you are having two phases. Octanol and water they are immiscible, and so you have a two-phase system, and that you put a substance into this mixture. Now, if you mix the mixture and place it down, there's going to be separation of phases. And when you have the separation of the phase over time, the octanol is going to separate from the water. Then you can take the upper layer, which is the octanol, and analyze it and see the concentration of the substance in there. And you take the water as well and analyze it to see if some of the substance is also in water as well. and then you strike a ratio which we refer to as a partition coefficient. Perhaps you would have done it in one of the practical courses in chemistry, where you do calculate partition coefficient of a substance, which is the ratio of concentration of the substance in a how do you say, in a water insoluble phase. So let's say if specifically we are using octanol, and so we say concentration of the substance in octanol over the concentration of the substance in water, which is what's that ratio is what we refer to as a partition coefficient. But this partition coefficient is also expressed many times as log P, which is log base 10 of that value of that partition coefficient which you have calculated. You come across it in pharmacology, in pharmaceutical industries. If you work in pharmaceutical industries, a lot of times is log P is being referred to see how much of a substance will be, say, dissolved in water or in octanol. And so the higher the value of log P, the more lipophilic the substance is. And the lower the value of log P, the more soluble the substance is in water. So you have this system of determining whether a substance is hydrophilic or the substance is hydrophobic. This way, it can be predicted that when the substance gets into the system, it can easily penetrate through cells into the cell, or it cannot penetrate through cells, so it will just be in the aqueous extracellular space. All right. Now, another thing, so you notice that we've moved from absorption. Now we've gone into distribution. Now, a major term which is used in the expression of distribution of substances in the body is what is referred to as volume of distribution. That is, when substances get into the body, there are two major things that happen. We did look at it even right from the beginning, that you can have substances binding to plasma membranes, plasma proteins. I mean, those substances that have such affinity for blood proteins will bind to blood proteins. And then you do have some that have less affinity for blood proteins, which will mean that those ones can easily move out of the bloodstream and get into the tissues. Now, when we say volume of distribution, what do we mean? Here too is ratio of the concentration of the substance in the bloodstream compared to, so the ratio of the substance in the bloodstream to the ratio of the substance in the tissues. So you take a look at the fact that, so in the first case where you're looking at the volume of the low volume of distribution, what you see is that you have high blood concentration of the substance, which is, so if you see the area being black, it means there is more of the substance there. So in the cardiovascular system, you see that the concentration of the substance is high, which means the substance is having more affinity for blood proteins, and so it remains in the bloodstream than in tissues. So tissue concentration is low. So in that case, you have a low volume of distribution. Now, what that says is that the substance will mostly get excreted out of the body, much faster than than getting accumulated. Because if the substance is having more affinity for the tissues, and of course, this is also a measure of accumulation of the substance in tissues. You can have intermediate volume of distribution where you are having averagely, like say 50%, around 50% of the substance having affinity for blood proteins, and so stays in the cardiovascular system, still in the bloodstream. And you have, say about half of it also getting, stored in the tissues as well. So this is like the spread of the substance, not only in the bloodstream, but also some of it also in the tissues. So in that stage, we say it's having an intermediate volume of distribution. Now, high volume of distribution, because this is much of concern, because as you can see, if the substance is having less affinity for blood proteins, but high affinity to be stored in the tissues, that will mean that the substance is going to have very serious toxic effects over a very long period of time, because that substance is getting accumulated in tissues and is going to be the end. And the more the substance is getting accumulated in the tissues, the more the risks of the toxic effects, and even the period that this toxic effect is going to last, because the substance is not easily getting excreted out of the body, where you have it in such high concentrations in the tissues. So that is a high volume of distribution. So that if you do, say, mathematically, say the ratio of concentration of the substance in the tissues to concentration of the substance in the bloodstream, of course, that value is going to be very high, and that is what is being referred to as the high volume of distribution. Right. So we move on to talk about how we represent these things with some kinetic curves. On the left side, we representing say the stages of absorption and elimination. Generally, absorption, yes, some distribution along the point, and then elimination with curves. That is to say that if you take on the vertical axis concentration of the xenobiotic, and on the horizontal axis you have time. So over a period of time, if a substance gets into the system, say at time 0, this particularly, so like on the left side, that is oral. Through oral entry, you see from the beginning, the concentration of the substance begins to rise, because as the substance gets into the system through the oral entry, it begins to get absorbed into the bloodstream. And so that is where you see concentration is rising steadily up to, say, a maximum point. And then at that point, you see that there is a decline of the concentration. What is happening in that region there is that excretion, metabolism and excretion is occurring. Even some distribution is occurring at the same time, because if you have a substance that is getting absorbed into the bloodstream, and you having distribution of the substance into various organs and tissues, in that case, there's going to be slightly some steady state, very brief, short, and then once you have the substance getting distributed and more, the concentration begins to fall, and then because that's a spread, and then the substance gets into the liver, where most of the metabolism is occurring, and excretion starts, where the substance now gets to be excreted out of the body, and gradually the substance gets excreted completely, eliminated. That is fully cleared out of the body. That is if you take just the first curve as a one-time dose assist the body is exposed to. That's what you experience in that case. But you don't know, maybe for repeated, you do have some repeated exposures. And so for such repeated exposures, is depicted with subsequent curves to see that substance gets up to a certain point where even, like say, in the process of excretion, then there is a dose at that point, and then the concentration rises again. And so you notice that that concentration begins to rise little by little, little by little, above the first maximum point for, say, repeated doses of the substance. You can also see some figures that are being shown. For instance, say area under curve, the AUC you see there is referred to as area under curve. It's a measure of the exposure of the body to, I mean, say the total exposure of the body to a xenobiotic. And you also see, okay, Cmax, I mean, that's highest concentration of the substance in the bloodstream. That is what is being shown there. And then Tmax, at what point the highest concentration is reached. You do have, say, half-life. I mean, at what time, the period within which half of the substance which has gotten into the body is, undergoes process of metabolism and excretion, getting excreted out of the body. That's a half-life of the substance. And you have some, let's say, average concentration of the substance in the body, which is to say that if you're looking at, the whole curve, this whole kinetic curve, at what point does the blood concentration reach, say, half of the maximum, and at what point does it reach, say, a minimum concentration that can have effect. And you have, if you take a distance between that minimum concentration that can have a toxic effect, the distance between that minimum concentration and the distance between, say, if it is this average concentration, or maybe the very top there, which can have toxic effects, for instance. In fact, if it is looked at in terms of therapeutic effects, then you'll understand that distance is referred to as a safety corridor. So, I mean, that is to say that if you take in a substance, the substance has a minimum concentration at which it will have a certain therapeutic effect, which it's supposed to have, and at a certain maximum point, it will also have, that therapeutic effect. But if it goes above that maximum concentration, then it begins to exhibit a toxic effect. And so the distance between the minimum concentration and the maximum concentration is what is referred to as a safety corridor of the substance. So that at all times, when doses, like dose regimens, are being developed, they are developed in such a way that you have concentration within that safety corridor, so that you don't have the blood concentration going above that safety corridor, which will then result into a toxic effect. So you have things like that. Then you have in, say, intravenous route of entry. If a substance is injected, what you notice is that immediately the substance gets into the bloodstream. So I mentioned earlier that, so there you don't have that process of absorption, because substance is directly into the bloodstream, and so the concentration is high right from the beginning, and distribution starts. So once distribution starts, you start to experience blood concentration levels dropping, and then you then will have, the elimination phase. Excretion is occurring all the way until the xenobiotic is excreted out of the system. So you see that the kinetic curves for oral, first of all, moves up before coming down because of that process of absorption into the bloodstream. But for intravenous, straight from the top, because the concentration is high from the beginning, and then it begins to come down. All right, so that's comparison between xenobiotics getting into the system by oral entry and through the intravenous entry. Right. So we've moved now from distribution. We have now come to metabolism, which is also referred to as biotransformation. So when you do hear these two terms, they mean the same thing. Metabolism or biotransformation in terms of xenobiotics or say drug substances, toxicants. Biotransformation is the same as metabolism. Now, different things can happen with xenobiotics as far as biotransformation is concerned. Now, first of all, you have metabolism of these substances divided into two phases. I mean, you have two major phases that you have different types of reactions that occur. Now, so here if you have a xenobiotic that gets absorbed into the bloodstream, you can either have the xenobiotic going straight to phase 2 without having any phase one interaction. It just goes straight to phase 2. And now, if you notice, you see that phase 2 is having a process of conjugation, which means you have some endogenous compound that binds to the xenobiotic, forming a complex, and then that complex gets eliminated out of the body. That is one thing that can happen with a substance. And the major reason for this process of, say, conjugation, or even the general reason for metabolism, why the body is trying to quickly metabolize the substance and things like that, is just because the body wants to do detoxification. It wants to make the substance less toxic and get it out of the body. It's protecting itself. And it's because most of these toxic substances are lipophilic. They can easily, get through the cell membrane into the cell, which is toxic to the cell, toxic to the whole system, because it will cause breakdown and fatality to the body. And the body is fighting it. And the body wants to quickly turn that substance into a hydrophilic form and get it excreted. And the major route of excretion is through urine. Urine is aqueous, so the substance has to be made hydrophilic in order for it to be excreted out of the body. Even if you think of it in terms of minor route of excretion being maybe through saliva or through sweat, all of these routes are having excretion by fluids which are aqueous. So you see a conjugation. We'll take a look at some of these specific reactions, and you see clearly that yes, a substance which is lipophilic is converted to a substance which is hydrophilic and getting excreted out of the body. Now, other things can also happen. You can have the xenobiotic undergoing phase 1 process, and a phase 1 process that is undergoing some metabolism which is not conjugation per se. I mean, other reactions occur, breaking down this substance, and the metabolites that are produced in this process can either have the activity of the substance getting lowered, or the activity rather gets increased. So there is some altered activity of the metabolite that is produced, or you can have the metabolite which is produced being non-active. That is to say that, okay, phase one has already detoxified the substance. The substance is no longer toxic with phase one reaction. It's a non-active metabolite. Or you can have the metabolite still being active, and that metabolite sometimes can even be more active than the substance itself, which, of course, even though the body meant to detoxify, it rather has caused toxification or toxication. And these things do happen. So we'll see some examples, and you have these metabolites then get through the phase two reaction, which is conjugation, bind to some endogenous compound, and then they can get excreted out of the body. So you can have these three different things that happen. So what we'll do is we'll take a look at what phase one is. I mean, look at what are the phase one reactions that occur. We can group them, and so we'll know the specific reactions that occur in phase one. And then when we come to phase two is easy. All of it is just conjugation. And so we just look at some examples of conjugation, and then we'll get to understand what it means. Conjugation only means binding of one molecule to the other. And so you have say a complex or some bigger molecule, which is more hydrophilic and can get excreted out of the body. So that's what we'll do now. We'll take phase one and phase two. Now, I mean, phase one reactions are separated into, three major, kinds of reactions. And you can even say two, because oxidation and reduction are always moving together. I mean, what you do refer to as redox reactions. These are phase one reactions. These are major reactions that occur with xenobiotics. And oxidation reactions include reactions like hydroxylation, decarboxylation, formation of some oxides, desulfurization, so substances that occur sulfur, you're removing the sulfur from them, dehalogenation, substances that have some halogens in them, like chlorides, bromides. You have reactions to remove these halogens from them. Oxidation of alcohols, because that's oxidation of aldehydes. And so these are reactions that occur with alcohol metabolism. And you see reduction reactions like reduction of aldehydes, some azo reduction, I mean, some hydration of nitrogens, and then you have some reduction of nitro compounds. And you can see all of these reactions are occurring in either the cytoplasm, I mean the cytosol, or microsomes. I mean, microsomes, think of it simply as endoplasmic reticulum, because the endoplasmic reticulum in the cell is that system of excretion in cells. So you have some small vesicles that are generated from the endoplasmic reticulum with waste products that get excreted out of the cell. And these are referred to as microsomes. So, but simply, you can think of it as endoplasmic reticulum. That is where these oxidation reduction reactions are occurring. And even the hydrolysis, so breaking down of, substances like maybe some lipids, which are like esters. So splitting of these esters, breaking down of some amide bonds. All of this is occurring also in the microsomes and the cytosol. So redox reaction, hydrolytic reactions, all of these are phase one reactions in xenobiotic metabolism. Now, In the liver, you also come across a major group of enzymes referred to as the cytochrome P450 superfamily of enzymes. And these are some isoforms of enzymes that carry out major metabolic reactions on xenobiotics. And out of these, you mostly hear of this cytochrome 3A45. This is the isoform of the cytochrome P450 enzymes that has a lot of metabolism of drug substances. And so what you see actually this light part of the diagram is showing the cytochrome P450 enzymes that have pharmacological relevance, where you see that the cytochrome 3A45 is responsible for metabolism of substances that have large molecular mass. They are lipophilic. They can be basic. So lipophilic will mean that they can easily penetrate through the cell. They are basic or acidic or neutral. So a lot of these drug substances have these properties. And so you have this cytochrome, the cytochrome P3A45, being the major isoform of the cytochrome P450 enzymes that are involved in the metabolism of xenobiotics. And then you have the cytochrome P2E1, the last one, or the first one on the left there. The first one is the one which is of much more toxicological relevance. Primarily because also a lot of these toxic substances, they are actually toxic even at very low concentrations, and those substances are small enough to penetrate through the cell. And when a substance is hydrophilic, it can easily penetrate through the cell, so it will cause some cellular toxicity. If it is if it is hydrophilic, it cannot penetrate through the cell, and it will just be in the intercellular space, but will be getting distributed to like being in the bloodstream, getting distributed into organs and causing a lot of toxic effects in different organs, things like that. But if it is lipophilic, it will get into the cell. So, but this experimentally, with research, this enzyme, the cytochrome P2E1, is of a major relevance to toxicological effects of substances, because it's that enzyme which is very much involved in the breakdown of toxic substances and getting them excreted out of the body. Then you have these other ones also. 2C9, 2C19, 2D6, 1A2. All of these are some isoforms of these enzymes that are involved also in the metabolism of different xenobiotics. Anyway, these are not things which you should say you will want to cramp into your head and things like that. A lot of times when you work with some of these things over a certain period of time, some of them stick. For instance, if you are into research into, say, maybe activity of, say, cytochrome P2C19, you get to know more about it over the period you are working on it and things like that. So, and you begin to know more of activity, the kind of substances they act on and things like that. So you don't try to cramp any of these things into your head. You just get exposed to them and know that you have a super family of cytochrome P450 enzymes, and different isoforms act on different classes of of xenobiotics. And that is enough of knowledge to have. And on it, if you go into toxicological research, you can build more on the knowledge with specific research results that you get out of it, and you get to understand it the more. All right. Now we can also take a look at briefly uh, the mechanism of action of these cytochrome P450 enzymes. And we did see that lots of this metabolic process is occurring in the endoplasmic reticulum. And so you see here, we say ER membrane, it means endoplasmic reticulum membrane. So the endoplasmic reticulum membrane, inside of the endoplasmic reticulum membrane, you have the cytochrome P450 enzyme complex. So this is an example of a cytochrome P450 enzyme complex with some NADPH cytochrome P450 reductase. And of course, we did enzymology, and so when you see NADPH quickly, you know that that's a coenzyme which is involved in electron transfer, which is in redox reactions. And you also see there FAD. Also, we saw it, the flavin mononucleotides or flavin dinucleotides, which are also involved in transfer of electrons in redox reactions. And so you see that's an enzyme complex. That's another thing which you see with the cytochrome P450 enzymes is that they are hemoproteins, meaning that they contain heme. Just like you see hemoglobin is having heme at the center, and you at the center of that heme, you have Fe2 plus ion, which is responsible for carrying oxygen. The same way you see these cytochrome P450 enzymes, they are also hemoproteins. So they have this ion at the center, which is also responsible for the activity of the enzyme. And then we did talk about active center of enzymes. Here, this thing is being shown as a catalytic chamber. It's just referring to the same term as the active center. Here is saying a catalytic chamber. And what do you see? You see there's a lipophilic substrate. That's the toxicant, the xenobiotic, that gets into the catalytic chamber. And there you see oxygen is being broken down with some action of water. There's some participation of water there. Water is broken down into hydrogen ion and a hydroxyl, and you have the hydroxyl binding onto the substrate, the xenobiotic that enters the catalytic chamber. And so you see the metabolite, it gets conjugated with that hydroxyl group. That's a hydroxylation process that has just occurred. So the metabolite will now become more hydrophilic, and then exits that catalytic chamber, and then it can get excreted out of the body. So I mean, this is just a very simplification of what the enzyme does. So this enzyme substrate complex, just depicting hydroxylation of a xenobiotic and making that a metabolite that is produced from the xenobiotic, more hydrophilic, that can get excreted out of the body. Now, we did also look at inhibition. They look at enzyme kinetics and inhibition. So this is where you can also apply that knowledge of enzyme inhibition in terms of how, you can actually use that to deal with intoxication in some cases. So here what you see is that you have inhibition of cytochrome P450 metabolic pathways, where you have you are saying that, yes, this is a cytochrome P450 enzyme. It's an active site that is being shown. Then you have a substrate, which is a toxicant that can bind onto that cytochrome P450 enzyme. And have a product, which is a metabolite, like what we just saw. Hydroxylation occurs, and so you have that product which will become more hydrophilic and can get excreted. And now you do remember we did talk about competitive inhibition, which you have a substrate, and then another compound which is say a structural analog of the substance that binds, competes for the active site. So you're saying that yes, the substrate is there, but the inhibitor comes to bind onto the cytochrome P450 enzymes and inhibits the enzyme from binding to the substrate. I mean, so that's what we learned with that competitive inhibition is also applied in this case in terms of um, intoxication of substances. So for instance, even though this example is not specifically with cytochrome P450 enzymes, this is with alcohol dehydrogenase. Maybe we looked at it, where you have methanol intoxication being managed by introduction of another alcohol, ethanol, because ethanol will bind to the same enzyme, alcohol dehydrogenase, but the metabolism of ethanol is less toxic than the metabolism of methanol, so that in that case, methanol is not able to get metabolized to say formaldehyde and formic acid and cause such adverse toxic effects, but will just get excreted unmetabolized. In this case, also with the knowledge of this competitive inhibition, you have some drugs that can be developed so that they can serve as inhibitors to some specific cytochrome P450 enzymes, the ones we just saw, which we know are specific to the metabolism of some specific xenobiotics, so that when they bind on the cytochrome P450 enzyme, then those substrates, which perhaps we know that those toxicants, when they undergo some specific metabolic reaction, they produce toxic metabolites, so that the inhibitor prevents the enzyme from producing those toxic metabolites. And so the xenobiotic can get excreted out of the body unmetabolized. You do also have non-competitive inhibition in these cases, not as common as the competitive inhibition, which we also saw, where you have the inhibitor binding to some allosteric site, which is different from the active site of the enzyme. So there's no competition here with any substrate, but upon binding to the allosteric site, changes the conformation of the active site of the enzyme, so that the substrate, the xenobiotic, is not able to bind onto the enzyme for the enzyme to have its metabolic activity on it. You do have these mechanisms which are explored in drug discovery and development. Drug discovery and development. You do have these kinetics of inhibition being explored in these cases. Now we move to phase 2 reactions. Phase 2 reactions, which we said were conjugation. They were all conjugation reactions. And you can see they are all occurring in the microsomes or the cytosol, the endoplasmic reticulum, or in the cytoplasm. That's where you have these phase 2 reactions occurring. You see major reactions like conjugation with glucuronic acid, which is referred to as glucuronidation. You have conjugation with sulfates, which you see as sulfation. You have acetylation. Acetylation, that's conjugation to an acetyl group, to an acetyl functional group. That is acetylation. You have some glutathione conjugation. I don't know if there's a term called glutathination. It's possible. These scientific words can easily be found. And you have methylation, which is conjugation of methyl group to the substance. So you have these different types of conjugation reactions, which are responsible for detoxifying xenobiotics and then getting them out of the system, by different forms of excretion. So we'll take a look at different conjugation reactions and see how this looks like. So we take this time we use the example of an endobiotic, which is bilirubin, because it's a common example where because you have red blood cells that are constantly getting broken down, through recycling, and you have bilirubin, or even in cases of anemia, you can have red blood cells break down, and the heme in these red blood cells, when they break down, they produce bilirubin. So in blood, you have bilirubin that can bind to the plasma protein albumin, and that then is taken into the liver, because it's flowing in the bloodstream, flows to the liver, and once it gets into the liver, you have, of course, the bilirubin getting separated from that albumin. So just bilirubin now is undergoing some glucuronidation. So in the liver, you have residues of glucuronic acid that bind onto bilirubin to give you bilirubin diglucuronide. You have an enzyme there, which is the glucuronyl transferase, which is acting in that process. So in the liver, that is where that process of conjugation occurs before you now have the bilirubin diglucuronide, which will be now hydrophilic and can flow freely. Goes to the bowel. First of all, gets stored in the gallbladder with bile, and when it gets excreted, bilirubin diglucuronide gets excreted through the bile duct into the digestive system, and so in the end, you have it getting excreted through fecal matter. Of course, part of it also can get excreted through urine. We can look at it much more, molecularly. On the right-hand side, you see that is the structure of bilirubin. It's just the ring structure of hemoglobin when it gets broken down into a straight chain structure with some oxidation at the sides with those blue oxygens there, and even some reduction with those red hydrogens on there. You have bilirubin, and bilirubin forms a complex with albumin in the bloodstream, flows into the liver. So now in the liver, you are seeing the reaction of conjugation of glucuronic acid with bilirubin. So the glucuronic acid residues are what are being shown red here, which are bound on the bilirubin residue, giving you that bilirubin diglucuronide. This then is secreted to the bile, stored there in the gallbladder, and from there, when bile is secreted into the duodenum, you have it getting converted by some microbial enzymes in the intestine into urobilinogen. You see, urobilinogen goes to the kidney, and in the kidney gets converted into urobilin, which then comes out with urine. So part of it gets excreted in urine, which is actually responsible for the yellow color of urine. Because constantly you have red blood cells breaking down and getting recycled, and so you have some amount of urobilin in urine, except that in cases of anemia, you have an elevated amount of this urobilin, and so urine tests are done, to test for bilirubin amount, but actually what is being excreted is urobilin. Same way, you have some stool tests that are done, so you see that urobilinogen gets converted also by microbial enzymes in fecal matter into stechobilin, and that stechobilin is what is responsible also for the brownish color, the natural brownish color of fecal matter. You do see stechobilin giving that color there. So that is a process of glucuronidation. That is a conjugation reaction, rendering the bilirubin, more aqueous and can get excreted out of the body. Because if bilirubin is not conjugated, just bilirubin on its own getting accumulated in the system can have some health effects. Particularly for children, it can have some toxic effects for some organs, even like for the liver. It can have such effects there. So the moment it gets conjugated with glucuronic acid, it's a form of detoxification of it. Or even some pigmentation issues with persons that are having things like jaundice, where they have this high amount of bilirubin, and you see it in the eyes being yellow, the skin pigmentation. So you have the effects in that manner. But that is if you are having bilirubin, the breakdown of red blood cells at such high rate that glucuronidation is not able to take care of all the bilirubin molecules, then you have such effects occurring. All right. Now we take some drug substances like antipyrin, and we see what are some of these reactions that we looked at that can occur on these drug substances. So, for instance, I've marked some specific portions of the drug so that we can trace what happens. We see that so number one itself, that is the drug antipyrin, and you see I've marked one methyl group there in violet and another methyl group there on that nitrogen with green, and then we trace it and see what happens. Now, if you look at the first reaction, on the left there, you see on that methyl group, you have some hydroxylation occurring, and so the metabolite which you find there is 3-hydroxymethyl antipyrin. So you see that's some hydroxylation occurring. That's a phase 1 reaction. And if you come down, you see that on the same carbon, that hydroxyl group there, that on that carbon, you now have oxidation, so that you have a carboxylic acid group. So that's an oxidation reaction that has occurred there. So you see a drug substance, when it gets into the system, it doesn't just undergo, say, one reaction. It undergoes several different reactions, so that different metabolites can be produced from the same substance. So even as we looked at those phase 1 reactions, phase 2 reactions, it didn't mean that a substance will undergo only one of those reactions, single reaction, and produce one metabolite, and that is all. No, as you can see from here, you see that the substances that are going different reactions and producing different metabolites. And you see here, looking at this methyl group on nitrogen, which is green, if you come to this, the third, the metabolite number 3, which is norantiparine, you see that methyl group is removed. So that is demethylation. Methylation has been removed, and the methyl group has been removed. In place of it, you have a hydrogen. That is demethylation. On the right-hand side, all these other reactions that you are seeing, yellow, yellow, yellow with the hydroxyl groups, clearly all those reactions are hydroxylation reactions. So all of these are, some phase 1 reactions: hydroxylation, demethylation, oxidation. They are all phase 1 reactions. Now, if we go to look at, let's say, codeine and morphine, which are also some drug substances, we see different reactions that also occur here. So if we take codeine, which is below here, codeine can undergo N-dealkylation. You can say N-demethylation, because the methyl group is an alkyl group. So specifically, you can say it is N-demethylation, because on this nitrogen, where you have that methyl group, the reaction goes into the formation of norcodeine, and you see that methyl group is removed. So that is a process of N-dealkylation, also a phase 1 reaction. From codeine to morphine, going up, there's a methyl group on this oxygen. So here you have the process of O-dealkylation. You see the difference? N-dealkylation means the methyl group is removed from nitrogen, but O-dealkylation means the methyl group is removed from oxygen, because the methyl group is bound there on that oxygen atom. This is some ether bond which is formed here, and you have that methyl group removed from it, so that you can see in the product you are having, in place of that, just a hydrogen. So you have a hydroxyl group replacing that methyl group there. Similarly, you have here too, from morphine going to normorphine, you can trace and see that this methyl group in morphine is removed. You don't see it here. What you come to see is just a hydrogen. So you have N-dealkylation occurring in that process. All of these are phase 1 reactions occurring with these drugs, codeine, morphine, which you see as some active ingredients of some painkillers, of some analgesics. Another, common analgesic, paracetamol. What occurs with it? With paracetamol. So you have the paracetamol and acetaminophen, and you see also undergoing different reactions. So let's take the reaction which is going up, and that reaction is glucuronidation. About 50 to 70% of paracetamol undergoes this glucuronidation. The reaction we just saw with bilirubin, this is happening with this xenobiotic glucuronidation. So you have glucuronic acid binding onto, so in place of that hydroxyl group in paracetamol, you have glucuronic acid binding on there. So that gives you a paracetamol glucuronide. And so what it tells us is that 50% to 70% of the drug paracetamol taken in just gets conjugated with glucuronic acid and gets excreted out of the body. And so it means you are left with another maybe 50 to just about 30%, which will be left if that can carry out any, um, therapeutic effect. But another interesting thing you even notice is that if you calm down, that is actually the pathway that will result into the therapeutic effect of paracetamol. And you realize it is only 1 to 2% of the drug which goes through that pathway. That you have, paracetamol, first of all, undergoing some deacetylation. So the acetyl group on that nitrogen there is removed. And so you now have just an amine group there. You have a para-aminophenol. And that para-aminophenol then undergoes, conjugation with arachidonic acid to give you the metabolites referred to as AM404, which is, you can look at it as arachidonyl para-aminophenol. That is actually the metabolites that is having the analgesic effect of paracetamol. I mean, almost most of the therapeutic effect of paracetamol is carried out by this metabolite, having its analgesic effect and anticonvulsant effect. The other pathways you see sulfation. So you see, in place of this hydroxyl group, you now have a sulfate group which is binding there, conjugating with that molecule. It's 25 to 35% of the drug undergoes that process of sulfation. Some oxidation reaction, 5 to 15%. So you see, this is a cytochrome P2E1. This the cytochrome one of the cytochrome P450 enzyme isoforms that we saw, and we refer to it as a major enzyme, a major isoform of that superfamily of enzymes having toxicological relevance. And you see it here, oxidation. And this oxidation results into the formation of a quinone. So you have this N-acetyl paraquinone. So in place of the hydroxyl group in paracetamol, you now see there is a double bond O there. A quinone is formed, and it is actually that quinone which is responsible for the toxic effect of paracetamol. Because this quinone can bind to proteins and form some protein adducts, which will then have some toxic effects. So, but you see, in the metabolism of paracetamol, this quinone is quickly conjugated with glutathione. So that's another conjugation reaction, and that complex can get excreted out of the body. But in cases of overdose, that is when you have this paraquinone being accumulated in the body and can form these protein adducts, which will then be causing some toxic effects. So you see, you do have paracetamol intoxication. Now, you have other drugs which are like, which are primary amines. I mean, these benzene rings with some side chains are, and then you have an amine group on them, and they undergo the process of N-acetylation, where you have, acetyl-CoA brings its, acetyl group comes to bind onto that nitrogen, that primary amine, gets it acetylated. The coenzyme A is release free. That's an N-acetylated metabolite. It's a common conjugation reaction you see with different kinds of primary amines, medicines, drugs. And even this has something to do with some genetic disposition of different individuals having, the enzyme which is responsible for this is N-acetyltransferase. And this N-acetyltransferase in different individuals can either, you have these forms which carry out fast reactions and forms that carry out slow reactions. So that in some persons that are referred to as slow metabolizers, some of these drug substances can get accumulated in the body, whilst persons that have that are fast metabolizers will have the drug substance metabolized very quickly and can get excreted out of the body, which brings to the subject of uh, personalized medicine. I think I talked about it the last time. Personalized medicine, where you need to have customized doses for different persons in order to have the therapeutic effects that is intended in different persons. Now, because we have some environmental chemistry people are among us as well, so as much as possible, we spread it. People who are doing maybe industrial biochemistry will have something from, say, maybe pharmaceutical side. Persons who also are doing industrial environmental chemistry should also have some little things. So we spread it a bit. So apart from all those, the metabolic processes which we have talked about, relating to those specific xenobiotics, you see those metabolic processes were occurring in the body, but you can have biotransformation, metabolism of xenobiotics occurring just in the environment. The substance does not get into a living body before the biotransformation occurs on it, but just in the environment, you can have these things also occurring. An example is this herbicide, which was used, decades before. Things, sometime in the 1940s, 50s, you had this herbicide developed, trichlorophenoacetic acid, but now it has been prohibited in all countries because over time it was identified that the substance trichlorophenoacetic acid undergoes hydrolytic reaction in the environment and produces this trichlorophenol, and the trichlorophenol can conjugate and form these dioxins. And dioxins, we did see dioxins are toxins which were produced from various industries, particularly from burning. The last time I think I did talk about dioxins being produced from burning of, indiscriminate burning with rubbish, like things happening at Agbubloshi, for instance, burning in industries, cigarette smokes, and these things have very serious toxic effects for unborn children, children in the womb. When they are, say, a pregnant woman breathes in any of these things, later that can result into some very serious problems for the child. And so this substance. So you see, this is where you have metabolites of xenobiotic getting even more toxic. And what are the kinds of reactions you see here? You see, first, hydrolysis. That's a phase 1 reaction occurring. This is just in the environment. And then also you have conjugation of the products, the trichlorophenol conjugation. That's a phase 2 reaction occurring in the environment to form this dibenzodioxin, TCDD, commonly referred to as TCDD. Now, you have another one, which is parathion. That's an insecticide. These two was an insecticide which had been used in the control of insects in like urban parks. But now is also prohibited. It's not being used again because it had also been found that in the environment it undergoes a process of photooxidation, gets converted into paraoxone, and paraoxone is also very, very much toxic to mammals like humans. Even though the parathion was used as a paralytic substance, acting on the enzyme acetylcholinesterase. But then when it gets exposed, when the parathyroid gets exposed to light, to sunlight, it gets converted into this very toxic metabolite. And so because of that, you have it now being banned. It's no longer being used. Another pathway that this also can undergo. So that photooxidation is a phase 1 reaction. Hydrolysis is also a phase 1 reaction where you see the formation of para-neutrophenol. And I mean, just this whole portion is broken off that ring to give you the diethyl phosphothiorate. All right. So these are examples in environments. Now, this is another interesting one, which occurred some decades ago, somewhere in, I think, one of the states of America. I don't remember exactly, maybe in Florida or so, that you had some company that was into production of into processing of nuts, and there were some phytosterols in the nuts that were being released into a water body, and downstream, those phytosterols caused the masculinization of some fishes in the river, so that those female fishes began to have some male features, began to behave like males, and became sterile. And so you see, that's a phytosterol you think is not toxic. I mean, this has some oils, sterol coming from, say, seeds, and the nuts which are being processed, and that waste product which you thought that was not toxic, but when they get released into the river, you have some microorganisms in the environment. But of course, this is occurring in a living organism. That is biotransformation occurring in a living organism. And then those phytosterols get oxidized. And so you see clearly that hydroxy group there on that sterol gets oxidized into that, cyclic ketone group. That's an androgen, testosterone. And then you have it, having such an effect of masculinization in the fishes. Now to the last end, to end it quickly with the process of, we've done so well because, I mean, to go over all these processes of absorption, distribution, and metabolism, and excretion over this short period of time, that's a marathon. Usually these things are done maybe different lectures. And so, but you see, because of the content of the course, if we want to do that, then we don't know when we are actually going to get over everything. But I think we need to get at things, just everything quick, little by little, and then we can go over the various things that we should go over, so you can have the best out of the course. Now, elimination can be by renal excretion. Renal excretion means you have excretion through urine. That's a major pathway. You can have excretion through stool, fecal excretion. We did talk much about these things already, so we'll not waste so much time. And I mean, by renal excretion, if you remember your senior secondary school biology, you also remember that, like, if you have a xenobiotic in the bloodstream, flows into the kidneys, and you have the building blocks in kidneys responsible for filtration of substances, is the nephron, and that nephron, you have this glomerulus, what is referred to as a Bowman's capsule. Secretion occurs, and then you have movement of the substance all the way to, say, collecting that, and from the collecting that, you have the substance, and these metabolites going to be stored in the bladder, and from the bladder, they get excreted through urine. I mean, so this is some, sort of a revision that you want to see. You want to understand how does the substance get excreted through urine. You also have excretion referred to as biliary excretion. These two we refer to it throughout in the lecture, that you have substances that get stored with bile in the gallbladder and later get excreted into the digestive system, into the duodenum together with bile. And then later they get excreted through the fecal matter. And that type of excretion is what is referred to as the biliary excretion through bile, or pulmonary excretion, which is for gaseous metabolites, exhalation, that is, coming from the lungs. And so it's referred to as pulmonary excretion. You also have excretion by the skin through sweat. We refer to it. That is referred to as dermal excretion. So I think one of you, who was that? Somebody asked a question about, I think, was it Christopher? Christopher talked about the hair. So you see what I was talking about, that you have the integumentary system being the skin. You can see that inside here you have the hair as well. The hair, the skin with its appendages. And so when you refer to dermal excretion, sometimes you also will consider the hair and even the nails, because they are considered as appendages of the skin. Then lacrimal excretion can have excretion of xenobiotics through tear from the eyes. I mean, when I made some example of, say, maybe tear gas, for instance, that is thrown, and then you have it having some irritation on the skin and then on the eyes. Tears have come into the eyes. I mean, that is quickly to wash off these things. Or even if you take some drops, some nasal drops, some eye drops, some ear drops, some of these substances by excretion, some of it comes through the eyes. All right. And then I think this should be the last one: salivary excretion and mammary excretion. So yes, some substances also get excreted with saliva, the metabolites or the substances themselves. And so you do have them getting excreted with saliva. And then mammary glands, and this is important in clinical trials where substances are being tested to see, is in lactating mothers, do you have the substance getting excreted in mammary glands, which can have effect on breastfeeding babies in that age. So these are major routes of excretion of the substance. So I thank you so much for your audience. And if there is any question, if there is any contribution, any comments, you can write in the chat or you can raise your hand. We still have like 6 minutes, so you can raise your hand and then I will enable your microphone, you speak, and then I respond to you. All right, so the floor is open.